this is Miss Litton, and this is my wonderful AP Biology Period 1 class. Say hi.
Hi.
You're so sweet. All right, so we are on Chapter 9, the cell cycle and reproduction. So we're going to talk about how cells divide, how you make more cells. And the big ticket items for this, okay, for this chapter is we are going to focus on the cell cycle. And when you talk about a cell cycle, it's its life cycle, basically. Like you have your life cycle, right? You're born, you crap your pants for a while, you eventually learn how to control that, and then you get to go to kindergarten or preschool, you go through elementary school, you go through middle school, which is super fun. And then high school, college, maybe start dating somebody, maybe you get married or whatever, have some children, you save your money to send them off to college, then you're a grandparent, and then you die. Okay, that is your life cycle. Okay, cells' life cycle, from the moment they are formed, they go through cellular division and they're formed, their life cycle takes place until they divide and form more cells, their life cycle. And so we're going to study the bulk of it. It spends its life cycle in interphase. This is interphase. And that's where it spends most of its time. And there are 3 parts to it, G1, S, and G2, and we'll talk about that. And then the steps of mitosis, the actual division. Mitosis is focusing on the division of the nucleus and where you could find the chromosomes when. That is technically mitosis, is nuclear division. Cytokinesis is division, there's a song there, division of the cytoplasm. Okay, and so we're going to go through this right here. Okay, the steps of mitosis and where we could expect the chromosomes at each time. That's also going to lend itself later when we talk about genetics and traits and how traits can get mixed up. We'll talk all about that. Now, we want to talk about the control of the cell cycle. It has checkpoints. There are 3 checkpoints we will go over. You have been all born or raised post 9/11, yes? No. Just a little bit before. See, let me tell you, you have no memory of, like, there are many times when I grew up and we were getting on a plane somewhere where literally somebody is throwing you out the door of the car and you know, like when I was in college, and you are running through the airport with your hard ticket in your hand, running through the airport, running all the way to the gate and like throwing them the ticket and running in. I mean, that has happened several times to me. Okay, when I was in college and just traveling around. You're just like, oh my gosh, we're going to miss the plane. You could get there 5 minutes before the plane's supposed to take off and run your way through the airport and just throw it, right? Now, oh my gosh, better be there 2 hours early. And if you've got an OCD parent, maybe 3. You never know. Going, right? And there are several checkpoints. Your parents can't go with you. If you're flying by yourself, your parents can't take you to the gate, right? No. So you have your first line of security is you have to even have a ticket to even really go anywhere in the airport, right? So first you have to have a ticket. Then they'll wave you in. Then you have to go through what? Security. And there you have to show your ticket, your ID, and anything you're going to take on is going to get looked at, right? And then there's a third checkpoint, and that is before you actually get on the plane, right? You have that third checkpoint, and that's how it is with the cell cycle, okay? And it serves a purpose because you don't want cells to be making more cells unless the source of the cells, the DNA, is good, because we don't want to make bad cells, and you need to have enough nutrition to provide for those offspring cells, so there needs to be the nutrients available. So there's certain checkpoints along the way. Now, if you lose it, without those controls, you can have things like what? Cancer. Cells growing uncontrollably, okay? And then that causes problems, bless you, for other cells. So this is, in a nutshell, what this chapter is about, okay? So first of all, when we talk about mitosis, it is a baby. Baby, where are you, baby? Okay? This is making more body cells. Yes. I am recording this. That's why I said, yeah, my first. That's okay. That was Gabby. All right, now, these are making more body cells when you talk about mitosis, and that if I wasn't talking about mitosis, what else might I be talking about? Meiosis. Now, this is how I remember this. Like, if you skin your knee, right? If you skin your knee, your two knees don't have to have sex with each other in order to make more knee cells, right? You know, like, what's going on? Yeah, I fell down. Okay? They don't do that, okay? It's asexual reproduction. So the way I remember this is my arms, my legs, mitosis. My arms, my legs, mitosis, okay? And that's asexual reproduction. When you do meiosis, you're getting ready for sex, okay? So say that with me. My arms, my legs, mitosis, as opposed to meiosis. I'm getting ready for sex. All right, perfect. Now, those are two different ways to make more cells. And we'll be talking about that process. That'll be chapter 10 for us. So mitosis is somatic or body cells. Now, at some point, like, I'm not growing anymore. I'm not getting any taller. Only thing I can get is what? Wider. I can get wider, but I can't get taller at this point. I have been done growing for many, many years. You are still growing, right? A lot of you are still growing, still getting taller. And you're creating more cells, but eventually, what's the plate? What do you not want to break in a bone?
The bone.
There's a certain part of the bone you don't want to break.
The middle.
No? Thank you. That was Maria. The growth plate. You don't, bless you. You don't want to break the growth plate because what can happen? That stops growing. And I had a student who showed me he had a terrible break in his growth plate, and I didn't. So my arms are the same length, but if I, like, it was literally like this. His arms were not the same length because of a terrible break. And where that can be really bad is if that happens in your what? Legs, because then that throws off your hips and walking and all of that. Because there are certain parts of your cell that are always generating more cells. Just like your bone marrow is still generating more blood cells. And so red blood cells and white blood cells, that part is still, but the rest of you, not so much. Okay? So, switch over here. When we talk about the cell cycle, let me get you caught up on your notes. Go to cell 9.1, cell increase and decrease. The cell cycle is from the moment the cell forms until its own division into two cells. And actually, we don't need to do anything yet. Okay, so take a look here. We're going to use this little black box to talk about what happens at each stage. I want to point out to you, all of this in purple is called interphase. And that's where a cell spends most of its time. So, in G1, okay, that part of interphase where a cell spends most of its time, it's only if it's going to divide is it going to go through this stage right here. Okay, in G1, the cell is growing. Now think about what happened right before G1. What do you see happening right before G1? You were two cells. So if you were one cell and now you're two cells, you're going to be what? Smaller. You're going to be smaller. So the cell is going to need to what? Grow. And you're going to need to make more organelles. Can you tell me what organelles are self-replicating? Mitochondria and chloroplasts. We don't have those, but yes, mitochondria and chloroplasts. Okay? So the cells are going to grow. It's going to increase in size. Okay? Part 2 of interphase is called S, and S stands for synthesis. And during this S stage, this is where you're going to make more DNA. Now, not all cells are going to enter into that S stage. You may just stop. And if you stop, you're going to go to this G0. We're going to talk about that in just a minute. But if you're going to head towards making more cells, you're going to need to make more DNA. So your DNA has to be replicated. And we're going to have a whole chapter just talking about how you double up your DNA. Okay? So you have twice as much DNA. Now we're going to move into G2. And in G2, this is where you're getting the proteins that you need in order to do both cell division and throughout the process of cell division and to be another cell. Okay? So G1, you're making more what? Organelles, and you're what? Growing. And in S, you're making more DNA. Okay? And in G2, you're making more proteins. Okay? Then you're ready to go through nuclear division. So nuclear division is mitosis. And there are different stages to it: prophase, metaphase, anaphase, telophase. Then division, that's division of the nucleus. Division of the cytosol, or what's referred to as the cytoplasm, is everything between the nuclear envelope and the cell membrane. That's called cytokinesis. And so that will form your two cells. So on your notes, go to stages of interphase. Most of the cell's life is spent in this stage. How much of its life? 90%. In G1, the cell grows larger, organelles increase. Blue, tell them what G0 is. Okay, and then S is DNA synthesis or duplication of the chromosomes. Another word for that is replication. And then what goes after the G2? Proteins are synthesized. Mitotic stage consists of two things. Mitosis is referred to as what? What did we talk about it? Nuclear division. Two nuclei contain the exact same DNA. And cytokinesis is division of the cytoplasm, forming two cells. All right, now this is my little animation I made for mitosis, the cell cycle. So get your bearings. What is the big black ring around the outside? Cell membrane. What's the little black dotted ring? Nuclear envelope. What are the colored things inside? Chromosomes. Now I want to tell you something about those chromosomes. The chromosomes, DNA only appears as chromosomes when you're getting ready to do mitosis. Okay? They don't coil up on each other in rod shapes unless you're actually doing mitosis right beforehand. It happens in prophase. Okay? So I put them in here just to make a point. You'll see that, okay, in just a minute. So, but they wouldn't appear as that. What do you think this is right here?
Mighty mitochondria.
Mighty mitochondria. Very good. Damn. Okay, what do you think this is supposed to be? Vacuole. What do you think these are supposed to be? They're abnormally large proteins. Okay, they would not appear that yeah. Now the cell cycle is like a ring, right? So you could point to any one spot. But where did we start in our cell cycle? G1. What did we say happened in G1? Growth. Okay, so the cell got bigger, and what else happened? Okay, so let's see if that happens. Did it? It did. Okay, then what comes after G1? S. What happens during the S stage? What happens during the S? DNA replicates. What does the S stand for? Synthesis. So let's see if that happens. Okay, so we double up on the chromosomes. Then what comes next? G2. What happens during G2? Okay, so now we made more proteins. Okay, what comes after G2? Mitosis and cytokinesis. Oh, look, it happened. Okay, so you get two cells. So on your cell cycle, not it.
Not it.
I would like whoever's not it to pass or play. Explain what you know about the cell cycle. Use this diagram to assist you.
I was not it. That's not
come back to me.
For G0, why don't they divide?
Because you don't need more of those cells. Now let's think about that for a minute, okay? Let me give you an example. Acromegaly. Acromegaly is when your bones continue to grow even when they shouldn't anymore. So literally, and it's only certain bones, bones of the face continue to grow. Bones of the hands continue to grow. And so that causes a problem because we're done growing. Don't keep getting a bigger, thicker face, right? And so that is that's just a probably uncomfortable position to be in, and there are other issues. But think of another cell where cells continue to grow. What do we call that? Cancer, right? So we don't want any more of those cells. You can have too much of a good thing. Red blood cells are important. White blood cells are important. What do white blood cells do for us? Kill infection. So it's like, can you ever have too much of a good thing? Yeah, it's called leukemia. Okay, when you have too many white blood cells, there's not enough room for other cells to do their business. Okay, so you would want to be able to, this is a perfect segue, because you would want to be able to tell those dividing cells what? Stop. People are investing millions and millions of dollars figuring out how to tell those cells stop. Okay, yes?
Why does cells like reproducing and replicating cause cancer in the first place?
We are going to totally talk about that today. That is what we are going to discuss. Good question. Yes?
Sort of related, but is cancer replicating of a bad cell, or replicating of just cells? Like too many cells or too many bad cells?
Usually if it's a cancerous cell, something's wrong with the control mechanisms, which would be in the DNA. So it's either not waiting for, let's say, a growth signal to start replicating. So that's broken if it does. Remember when we talked about the functions of proteins on the surface? What did we say? It could be what? A channel, carrier, cell recognition, receptor, enzymatic, and then we could also do cell junction, right? So some receptors, there's one, it's called the RAS protein, and this RAS protein sits in the cell membrane, and it needs a growth hormone in order to trigger it to make more cells. Well, if you have a bad RAS, okay, it's like stepping on the gas. Your cell, even though it's not getting a signal to replicate, it's like, let's go, okay? And it keeps dividing and dividing and dividing and dividing. And so that particular cell has bad DNA because it's making a bad RAS, which is getting stimulated even though it has no hormone telling it to grow. So there's cancer. When you have cancer, there's a couple of things that are happening there. You have a cell that's out of control. But the second thing is you have other cells whose job it is to patrol those bad cells and to take them out when you have cancer. And those genes that control that are called tumor suppressor genes. And they are the assassin who's supposed to monitor and go, okay, you're doing fine, you're doing, no, kill it. And every day you probably take care of a cancer cell. Every day you probably have a cell, and every day you probably take care of it. So when you ultimately get cancer, it's a combination of two things. One, you have a cell that's just going hog wild, who shouldn't be. And that's because a proto-oncogene has mutated into an oncogene. What kind of doctor do you go see when you have cancer? Oncologist, okay? So you have that issue, and the second issue you have is you don't have any assassin taking that bad thing out. You don't have anybody monitoring your bad cell. So when you get a combination of those two issues, out of control cell and no assassin to take out the out of control cell, that's when you have cancer. Yes?
When you have a really stage 1 of cancer,
yes.
You have a tumor, you're allowed to, but what keeps it from reproducing again inside of it?
Well, the different, for instance, testicular cancer, stage 1 is having cancer in the testes. Stage 2 is when it has metastasized, when it's left its own home base and it has moved to your lymph nodes here. This is stage 2 when it's in your groin, in the lymph nodes in your groin. Stage 3 for testicular cancer is when it moves into your lungs, and stage 4 is when it moves into your brain. So cancers typically have a certain pattern of where they go next. They have their regular journeys they go on. And keep in mind, your circulatory system and your lymphatic system, it's like a train station throughout your body. So when a cancer cell can get onto that train, it can now jump off anywhere else in your body and start to grow there. What keeps some cancer cells in check you've heard of benign and malignant. And we're not wasting our time, this just comes later, okay? Benign and malignant tumors. When they say, for instance, what is this month? Breast cancer awareness, okay? So you want to check to make sure you don't have a tumor, because you could have a tumor and you could be aware of it, you could feel it in your breast tissue or your testes, gentlemen, and when you feel it, you could be like, oh my gosh, you know, there's something in there. But it can be encapsulated because it hasn't got a blood supply to give it nutrients, because it's not going to replicate unless it has nutrients available to it. That is prevented. Your cell will not replicate. But if it gets a blood supply, because you know what tumor cells do? They're the dirtiest of dirty deeds, because they actually do something called angiogenesis. They send out chemicals that go, here, little blood vessels, come here. I've got some candy for you. And the blood vessels are like, what? Okay. And then they grow towards the tumor, and they dock in on the tumor, and then the tumor goes, yes. And now it has a nutrient supply, and it starts to go farther. Sometimes tumor cells are encapsulated, like skin. What am I trying to think of? Like a melanoma or something like that. Okay? So you're like, oh, I got it. You check for irregularly shaped moles or patches of dry skin. I don't mean like, oh, I wash my face too much and I have a dry patch. I'm talking about like skin is kind of flaking off there and it looks dry. It can happen on your faces a lot as you age, like my age, because you've been out in the sun, right? And so what happens is those areas, if you catch it early, you scoop it off and it's gone. But if it stays there too long, it can gain the ability through mutations to digest through the basal lamina, the layer that separates your skin from all your other stuff. So if it gets through that basal lamina, it can get into a blood vessel, and that's when it metastasizes, and that's when you're in trouble, where you have a hard time controlling it. You'll hear about when they remove growths or tumors that they'll look for the edges. Did we have clear borders? And they're looking for clear borders that don't have any cancerous cell, and they'll say, I hope we got it all. And they will literally, during their surgeries, send cells to the lab to have them check to see if they're clean, because they want to keep going until all the cells show up clean, and hopefully they got it all at that point, because they don't want it to spread to other areas. Okay, does that make sense? Tell your bio buddy what's one new thing you've learned just now in this little discussion. It's going to come up. Everything I said is going to come up again. Okay, when we go and talk about cancer, we need to be sensitive in the room, okay? How many of you know somebody in your family or know somebody who's been impacted by cancer? Okay? So for some of us, it's near and dear to our hearts. It's like I've had this presentation for a while. When we get to the cancer talk, scroll down right here. This is me with brown hair. So that's my mom, and we were up in Stanford, and she was there for a long time. So I mean, it's near and dear. So when I talk about it, even now, I get a little teary, because she was one of my best friends, you know, and it's been almost 10 years. So we have to keep in mind when we talk about these things, we could have people sitting right next to us in class right now who have a family member, an aunt, or an uncle that's going through this and dealing with this right now. So just be sensitive in how you talk about it, and as we discuss it, I just want to bring that up. And for those of you that are thinking about it, I just want you to know, I know that it's difficult, okay? Now, let's talk about the basics about control. So number one, your cell cycles are controlled by both internal and external signals. The signals that the cell is receiving internally and externally. So that's the first thing on your notes, controlled by external and internal signals. A signal is a molecule that either stimulates or it inhibits a metabolic event. And an example of an external signal would be something like a growth factor that's received at the plasma membrane, like I mentioned to you before about the RAS. And I don't know if this link is still good. Let's see. Come on now. Here we go. Let's take a little listen.
Replication of animal cells usually occurs only when specific growth factors are present. There are many types of growth factors that can be produced in a body to stimulate cell division. If the right growth factor is present for a given cell type, it will match up precisely with a specific cell surface receptor and bind to it. The binding of a growth factor to a receptor starts production of signals within the cell. Typically, this within-cell signaling system is a protein kinase cascade, a series of reactions where phosphate groups are passed along to different regulatory proteins in the cell. Ultimately, the cascade ends with phosphorylated proteins entering the nucleus and interacting with regulatory proteins there. The typical result is that some specific proteins are freed to activate genes for cyclins and cyclin-dependent protein kinases, CDK. The cyclin and CDK proteins then act to stimulate cell division.
Okay, so this would be a great example of reception of a signal and then signal what? Transduction and then this response. So it's like this triggers this, triggers this, triggers this. Now CDK, cyclin-dependent kinases. Now you just heard that phrase. What do you think of when you hear just those cyclin-dependent kinases? What are you thinking about? It's an enzyme. It's an enzyme that's going to stimulate cell division. Now this enzyme is obviously dependent on what? Cyclin. So as the cyclin level rises, it will trigger this enzyme to do what its job is, and that's to stimulate the cell cycle. These are all points of control along the way where you can either stimulate that cell or inhibit that cell. And you're going to be reading about that too in your highly suggested reading and thinking. Okay? So on your notes, your example, I just put growth factors. Okay, so let's talk about the checkpoints. So there are 3 different checkpoints, and the one that seems to be the most critical is the G1 checkpoint. And that makes sense to me because what follows G1? S. And what are you going to do in S? Make more DNA. So you don't want to spend your time making more DNA and wasting all those nucleotides if you're not planning on going through the process of cell division. So let's make sure everything is good. So at this point, you're going to check and make sure, and you're going to stop. And a key gene on this is called P53, and we're going to talk about him in just a minute, okay, in G1. And if you pass this checkpoint, then you can move into the S stage. And then in G2 checkpoint, this is, let's see if we replicated the DNA okay. If we made a mistake in our DNA replication, we want to stop and we want to repair it, because if it's not done correctly, we could have problems later, okay? And then again, you see this word apoptosis, and there was apoptosis here if it's not going well. What is apoptosis? Yeah, cell death, cellular suicide. Okay, so if we can't fix this problem, I'll fix this problem. We'll just commit suicide. Okay, that's how it's solved. And then partway through mitosis, you're doing nuclear division. Let's check. Everything going all right? Are the chromosomes separating appropriately? Because if not, we need to stop. Okay, and then let's watch this one. Watch me. Watch me now.
The process of cell growth and division in eukaryotes is called the cell cycle. This cycle is divided into phases based on what is happening in the cell at a given time. A cell grows during the G1 phase. During this phase, there is a chemical checkpoint that controls whether the cell will divide, delay division, or enter a resting stage. When conditions in the cell are right, the G1 checkpoint will be passed and the cell will enter the synthesis S phase. During the S phase, DNA replication occurs so that future cells will each have a complete set of the genetic instructions in the DNA. After DNA replication is complete, cells enter the G2 phase, where they continue to grow and prepare for cell division. At a checkpoint in this phase, the success of DNA replication is assessed. If all is well, the cell enters the mitosis, M phase. During the M phase, a complex series of events moves the DNA so that a complete set of genetic instructions will be sent to each daughter cell. The process of mitosis is assessed at a checkpoint during the M phase. Once this checkpoint is passed, the cell will complete mitosis as well as begin the cytokinesis C phase. Part or all of the C phase actually overlaps with the later parts of mitosis, so it is not a distinctly separate phase. During the C phase, the cytoplasm of the cell is divided, and two daughter cells are created from the original cell. When this process is finished, the daughter cells enter the G1 phase, and the cycle is complete.
Okay, at this point, you should be able to talk about the cell cycle without your notes, because we've gone over it several times, and the checkpoints. So whoever talked last time, you're hush-hush. Other bio buddy, talk through mentioning checkpoints, and correct them if they need a little help, but keep your eyes off your notes. See if you can just discuss it. Go ahead. Okay, I think I owe you some notes on that, yes? Okay, so your notes on that for checkpoints, the three, little letter I. G1 checkpoints appears to be the most critical. The cell will remain in G0 if it does not receive the signal to move into the, what follows the G1? S. Good. Number 2, cell check to see if the DNA is damaged or can't be repaired or chromosomes are not lining up correctly. And apoptosis if they don't pass one of the checkpoints. Apoptosis if they don't pass one of the checkpoints. Now, let's talk about these cyclins. Look in your picture, find the cyclin. Who does the cyclin bind to? Here's the cyclin, it's in blue. Who's it binding to? The CDK. Okay, the cyclin-dependent kinase. Now, this is an M cyclin that you would need in order to enter into mitosis in order to leave the G2 checkpoint. You all right with that? Okay, so on cyclins, these are proteins that control the cell cycle. They must be present, because they will build up, to move into the S and the M stages. Kinases, what do we want to put in parentheses? A kinase is a type of what? Enzyme. Good, enzymes. And they are activated by cyclins. Activated by cyclins. They're called CDKs, cyclin-dependent kinases. When a cyclin deteriorates, the kinase becomes inactive. So if M cyclin builds, it builds up enough to trigger CDK, okay, and then you will get a product from this that allow you to move into the next stage. Now, an example of that, okay, and that would be your next one right there, your example, okay, without me explaining this one, have other bio buddy, whoever's turn it is, work your way through explaining, look what's coming on here, and then work your way around. See if you can do it without me. Go ahead.
Cyclone binds with enzyme, creating the cyclin eventually degrades, and then the cycle continues with CDK.
Okay, so on this one you have the cyclin binds to kinase producing enough of the product, MPF. Now, two different names for it. Okay, I already gave you the maturation promoting factor that's in your notes, but another way to think of it is the mitosis promoting factor. That would go in your highlighted area. So, cyclin hooks up with CDK, you start producing MPF. Okay, if enough of it is there, you will move into mitosis. It will push the cell cycle through G2 and into the M stage. Later in the M phase, cyclins begin to be destroyed to stop the M. Okay, so they'll start to degrade, and okay, we're done. We're ready to move into the next stage. Now, this is more closely linked to reality than what I've been showing you. You're not learning this is triggering this, which is binding to this. You're not learning all of that. The details are pretty specific. If you look at this, CDK cyclin, complex of the direct functions regulating at CD3 with cyclin, okay? So you see the point here. There are things in here you are going to learn about. E2F is going to be one of those ones that you're going to talk about. And that's going to be in your highly suggested reading and thinking. And it focuses on how critical the G1 stage is. So you will have some reading about that. Okay, and then last but not least is apoptosis, cellular suicide. Keeps your numbers healthy, prevents tumors, it is programmed cell death. Critical one, and you'll be reading about this as well, is that P53 protein, which is coded for by the P53 gene. And basically, it makes some enzymes, some caspases, who will kill off the cell, kill it off. So on apoptosis, it's defined as programmed cell death. And that is a balance between mitosis and apoptosis, and it helps maintain the normal level of somatic cells, prevents a tumor from developing. And then I gave you the death caused by. All right, and here are some questions for you. Let's get some hundies. What's the number again, you guys?
72.
Yeah, 72 number. Let's get some hundies. After we finish these questions on this, do you want me to put a stop on this section again? And okay. Okay. Okay, Mama. This to me, you should be 100% on, yeah?
Yes.
Oh, yeah, you're right.
Oh my gosh.
All right, here we go. Here, let me click off and come back. It'll register it. That's it. This is clearly a hundy one. D, mitosis. No. The DNA is not replicated. What happens during mitosis? Nuclear division. Mitosis is all about nuclear division. Sheldon Cooper. Took you 42 seconds to fail. So sad.
Yeah.
What happens during?
The cat.
Okay. Guys, you know you have your notes in front of you, right? Okay. And we've gone over this, I want to say 4 times, minimally, conservatively 4 times. These are not mistakes we should have. Show your clicker to your buddy right now. Chastisement or praisement. G1, more organelles. G1, more organelles. Good job, Schrodinger's cat. Yes, thank you. Wait, sweetheart, turn the pass to the right of the phone. Let's go, people. Make a commitment. Maybe we just went over this maybe like, I want to say 40 seconds ago. During what stage is there nuclear division? That would be what? Mitosis. Show your clicker to your bio buddy right now, chastisement or praisement.
Yes, it does.
Okay, so there's 1, 2, 3, 4 of you that should be feeling shame right now. Okay? Shame should be heavy. Okay, shame. All lowercase. Holy crap on a cracker, let's go. Oh, not G0, apoptosis. If it doesn't pass the checkpoint, now, G0 could be a normal part where you're not making any more of those cells, right? So my bone cells right now are in G0. My osteocytes are in G0 because I'm not growing anymore, right? So that doesn't mean they need to be committing suicide. I just don't want them to grow anymore. Okay, so that would be, if you don't pass the checkpoints, then it would be apoptosis. Okay, and I believe that is the end of the questions on that section. Okay, so we'll stop it right here. Be smart.